So the next macromolecule type we're going to talk about are the lipids. So lipids are really defined by the way that they interact with water. So the special characteristic that lipids are going to have is that they're going to be hydrophobic. So they're not going to interact with water, and in fact, they're going to repel water. This is because our lipids are mainly going to be made up of just carbon-hydrogen bonds. So we call them hydrocarbons mainly. And bonds between hydrogen and carbon, because hydrogen and carbon have pretty similar electronegativity, are non-polar bonds. So because we have these non-polar covalent bonds, these molecules are considered non-polar, and non-polar things are not attracted to water, they repel water. So this is different than our carbohydrates, where we saw a lot of oxygen and therefore a lot of polar covalent bonds. So our carbohydrates were polar molecules that are going to be hydrophilic. Now, lipids have a number of different functions biologically. They can provide insulation, so fat is going to be a lipid. They're going to provide long-term energy stores. They're a really efficient way of storing energy in chemical bonds. We'll see that they form really the basis for our cell membrane, so they're going to be the most major component of a cell membrane. They're going to serve as building blocks for some of our hormones, and they're going to, in biology, provide protection for an organism from things like water, wind, and temperature, because they're good at repelling stuff. So there are four really types of lipids. We have our fats and oils, we have phospholipids, we have steroids, and we have waxes. We're going to talk about each of them so that we know how to recognize each different type. So fats and oils are mainly going to come in the form of triglycerides. And a triglyceride is made up of glycerol, glycerol backbone, and fatty acids. So let's take a look. Okay, so glycerol here, actually on its own wouldn't really be a lipid. We see these hydroxyl groups, but it's going to form a backbone to attach these long hydrocarbon chains. So these long hydrocarbon chains made of just carbon and hydrogen. See, we don't see any oxygen in these chains. Those are our fatty acids. So they attach on to a glycerol molecule, 3 of them to 1 glycerol. So when we're looking for functional groups in our triglycerides, where we're really going to look is at the end of our fatty acids. So at the end of a fatty acid, we're going to see, at the end that attaches to the glycerol, we're going to see a carboxyl group. So our carboxyl group here is our carbon, double bonded to an oxygen, double bonded to a hydroxyl group. And I highlight here in the top right corner that a carboxyl group is different than a carbonyl group. So a carbonyl group was just a carbon double bonded to an oxygen. Here we have a separate word, carboxyl. Okay, carboxyl tells us that we have this hydroxyl group in it. So double bond to oxygen, bond to a hydroxyl group. Okay. So we have 3 fatty acids attached to a single glycerol molecule. And our triglycerol, or triglyceride, then is going to look kind of like a flag with these 3 fatty acid tails coming off the side. Fatty acids can come in basically 2 different shapes. So we're going to have saturated and unsaturated fats. And that actually refers to the number of hydrogens that we're going to see in that hydrocarbon chain. So in a saturated fatty acid, we're going to see single bonds in our carbon backbone. See, we have all these single bonds. And each one of these carbons is going to have a hydrogen coming off each side. So it's going to be bonded to 2 hydrogens. In a saturated fatty acid, because we have only single bonds between our carbon molecules, this means this chain is holding the maximum amount of hydrogen it could possibly hold, which is why this is a saturated fatty acid. So what it's saturated with is hydrogen. Now, the fact that it has all these single bonds actually means something for its physical shape. So when we have these single bonds, we're going to have really a sort of straight linear molecule. And this straight linear molecule is really easy to pack together tightly. Because we can pack it together tightly, this means it's going to be a solid at room temperature. So some examples of saturated fatty acids would be like butter, the fat part of meat, things like that. By contrast, we have our unsaturated fatty acids, which are going to have a double bond in the carbon backbone. And this double bond in carbon means that we don't have as much space for hydrogen anymore. So remember that carbon can only take 4 bonds. So if it has 2 going to this neighboring carbon and 1 going to this neighboring carbon, it can only make 1 more bond. So usually with the single bonds, we can have 2 hydrogens. Now we can only fit 1. Okay, so that makes this an unsaturated fatty acid because it's not holding the maximum amount of hydrogen that it possibly could. If we have 1 double bond, we'd call it monounsaturated, mono meaning 1. If we have multiple double bonds, like say we had another one over here, another one over here, we'd call it polyunsaturated. So when you're looking at a food label and it labels saturated fats, what it was telling you is those linear fats, where we just have those single bonds, they're thought to be more unhealthy. So that's why our food labels tell us which type of fat there is in that food. Okay. So our unsaturated fatty acids are actually going to be liquid at room temperature. And can you think about why that might be? Yeah, so because we have that double bond, we're going to have a bend in the molecule, actually. So we're going to have a bend. We're not going to have this straight molecule that can pack so tightly anymore. So we're going to have more space in our collection of fats, or in this case, oils. And they're going to be liquid at room temperature. Now, this takes us into the topic of trans fats. So trans fats are going to be a very specific type of unsaturated fat. Okay, so when we were talking about isomers before, we talked about the fact that we can have different shapes of a molecule even when we have the same chemical formula. When we're talking about trans fats, what we're talking about is the position of the hydrogen on these carbons that are participating in the double bond. Okay. So if they're on the same side of the double bond, here we get a bend in the molecule. Because they're on the same side, we call this a cis configuration. So this would be a cis fat. So cis fat is bent. This fat is going to be it's going to be an oil. It's going to be liquid at room temperature. It's not packed as tightly. And from a health perspective, these are thought to be more healthy. So these are really your natural oils. Now, trans fats are actually usually made through food processing. And in a trans fat, we have this double bond. So it is an unsaturated fat, but these hydrogens are on opposite sides of the double bond. So trans, we remember, means opposite. Okay, so because they're on opposite side of the double bond, it's going to behave much more like a saturated fat because it's just going to be in this single line, this straight line again. So our trans fats, like our saturated fats, are thought not to be so good for us. Now, I said before that our lipids are really united in the fact that all of our lipids don't interact with water and actually repel water. So now that we're looking at the chemical composition of these fats and oils, can we tell why that is? So when we're looking at all these carbon-hydrogen bonds, these carbon-hydrogen bonds are all nonpolar. So if you look at this fatty acid, the only place you could possibly have any polarity is in this carboxyl group, right? And then when we create this triacylglycerol or this triglyceride, we bond that onto glycerol, and now it's actually going to be hidden behind these carbon-hydrogen bonds on that glycerol backbone. So when water comes up, those slight charges on oxygen and hydrogen come up to these carbon-hydrogen bonds. These are nonpolar. There's no charge anywhere on these bonds. And so that's going to repel the water. There's nothing there to interact with those little charges on the water. Okay, so the next type of lipid that we're going to talk about are the phospholipids. Now, phospholipids, really honestly for this class, are going to be the most important type of lipid we talk about because phospholipids are going to be what makes up our cell membranes. Okay, so when we're thinking about functional groups, we're going to look for in phospholipids. This phospho part, this is really telling us that we're going to have a phosphate group somewhere in it. So this is going to be like an addition to our lipid part of the phospholipid. And let's take a look. Okay, so our phospholipids are made up of this phosphate group attached to a glycerol backbone, then attached to 2 fatty acid tails. So when we were looking at triglycerides, our fats and oils, we had 3 fatty acid tails. Now we just have 2. We also have this phosphate group. Okay, now a phosphate group is actually going to be polar. So our phosphate group is going to interact with water. So we're going to call this phosphate part of the phospholipids. This is our polar head. And the polar head we're going to call hydrophilic. So this phosphate polar head of the phospholipid is hydrophilic because it carries slight charges, right? So immediately we can actually see a negative charge here on this oxygen in the phosphate group. So this is going to attract water molecules. In addition, this phosphate group might have even more charged and polar chemical groups attached to it. So this phosphate group is going to orient towards water. Now on the other end of the phospholipid, we have these fatty acid tails. And we already saw fatty acids, right, when we were thinking about our fats and oils. We know that our fatty acids don't interact with water. Our fatty acids are nonpolar, and so our fatty acids are hydrophobic. So these fatty acid tails in our phospholipids are going to orient away from water. They're going to repel water. They're going to not interact with water. There's a special word for a molecule that has these 2 parts where one side of it is attracted to water and the other side repels water. So that word is amphipathic. Our phospholipids are amphipathic, and that just means that they have a polar end and a nonpolar end. So they have a hydrophilic end, this part, right, that's attracted to water, and they have a hydrophobic end. So the hydrophobic part here is our fatty acids. Now, a really cool thing about phospholipids is actually that if you have a collection of phospholipids and drop them in water, they're going to immediately form specific shapes. So just the fact that they have one end, this polar head, this phosphate group part, that is attracted to water, and at the other end they have those tails, right, these non-polar fatty acid tails that are hydrophobic. This means that if you put them in water, they're going to spontaneously move so that polar heads face water, and those fatty acid tails, they're running away, they're trying to escape from the water. So those fatty acid tails are going to point towards each other and protect themselves from the water by using the phosphate heads, those polar heads, to kind of like block the water. So one of the shapes that these phospholipids are automatically going to form in water is a membrane bilayer. So if we have 2 phospholipids line up across from each other, we're going to end up with so say we're in a water-based medium, right? So we have this pool of water. We're going to point our polar head towards the water. We're going to have our fatty acid tails going in the opposite direction. Now there's still water over here, right? So in order for those fatty acid tails to avoid that water, because they repel it, right? They're afraid of it. We're going to end up with a second phospholipid. Its fatty acid tails are going to come in nice here next to the fatty acid tails from the opposing phospholipid. So then they're grouped together. They're both nonpolar, so that's fine. They're attracted to each other. And the phosphate group, that polar head of the second phospholipid, is going to protect us from the water on the other side. So that's what we see happening here. Here we see our polar heads on this side, our tails in the middle, and then the second layer here, we have the tails facing into the middle of the membrane and the polar heads here. Now this is going to be really important when we talk about cells, because the outside environment in an organism is often going to be made of water, right? Like our blood, interstitial fluid, mainly water. But the cytoplasm inside the cell also has a lot of water in it. So we're going to need something polar on both sides, something that's hydrophilic, something that's attracted to water, pointing towards the outside environment and pointing towards the inside environment. So that's why we have this double layer. And we'll talk a lot more about membranes in the future. So our last two types of lipids are going to be our steroids and waxes. We're going to spend less time focusing on these, but you should be able to recognize them. So a steroid, which is going to form a lot of our hormones, although there are also some protein hormones. Our steroids are going to be characterized by these 4 rings pressed against each other. So that's really how we're going to tell we're looking at a steroid. We're going to have one ring here, press directly against it another ring, press directly against it another ring, press directly against it another ring. And why I'm emphasizing that they're rings pressed against each other is because if you're trying to identify a macromolecule, right, a good way to identify a carbohydrate is that we have a chain of rings all linked together, right? So there could potentially be 4 of them. But the difference here between the carbohydrates and the steroids is that these sides are directly in contact, so we can see they're connected in multiple places. Whereas if you flip back and take a look at some of our carbohydrates, all our carbohydrate rings have a covalent bond to oxygen in the middle and another covalent bond out to the ring. So the rings in carbohydrates are more separated. They look like circles holding hands. Our steroids are all pressed against each other, and there are always, always, always going to be 4 rings pressed together, so like this. This part of the steroid can vary, so you could see a number of different things here. So you're going to recognize this special 4-ring structure to recognize a steroid. Our waxes are basically just fatty acid chains floating around on their own. So they're like our fats, kind of, right? We recognize the fatty acid structure. So it's just carbon, 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 carbon. Remember, every point in a diagram like this is a carbon. Now, there's nothing listed, so we assume this is a carbon bonded to a hydrogen here, bonded to a hydrogen here. So our waxes are going to be nonpolar, like our other lipids, but they just don't have any other component parts. So they're really just a long fatty acid chain, and they might be kind of bent and linked together, but they don't have a backbone or anything. And that's it for lipids.